Today we have the honor to award both the Early Career Award and the Frits Hobels Award, and I'm going to introduce the Early Career Award winner. So a bit more about Marco and why he was selected. So there are some rules. You have to be under the age of 35 or have defended your PhD in not more than 7 years ago. So yeah, he was fulfilling these two criteria. On top of that, both nominators told us that he was very active in EEMGS as well as national society meetings in Croatia. Here is some of the feedback that we got from the award committee. So they said we should consider him even though he was the only one that got nominated. And this is kind of a summary of what they thought about his CV and his achievements. Well, Marco, again, congratulations with this award. You clearly deserve it. And now the floor is yours.
Good afternoon, everybody. Thank you, Sabine, for this nice introduction. When I first received email stating that I was going to be presented with this award, I really thought to myself, okay, this is like winning an Oscar, and I was really thrilled and excited. And today I'm really happy and quite honored to be able to be the awardee here. I definitely have a lot of thinking to do, but I will do that after my talk. So I would like to go straight to the presentation now. Okay, so I will start here. Yeah, unfortunately we are not in the portal right now, but I wanted to keep this picture since it should have been like the highlight of the meeting. It represents bridge. And that was also a bit of a motivation for my title. So bridging the occurrence and effects. Okay, I'm going to talk about cytostatics again. Unfortunately, it won't be boring. But to start this presentation, I need to go back to my student days when I was working on the toxicity of pesticide DDT. And back then, I got more into this Rachel Carson's Silent Spring and found this interesting quote. And ever since I got employed here at the Institute in this group that I'm currently part of, we were trying to detect as much as possible pollution spots, and we were trying to minimize these alterations we humans do to the nature. So apparently I'm going to talk about anti-cancer drugs in the environment, and let me just briefly walk you over the current statistics we have. So in the 2018, the number of newly diagnosed cancer cases was slightly over 80 million. And in the same year, more than 9.5 million died of cancer. So if we take current rates and the expected population growth, by the time of 2040, we expect to see over 29.5 million new cancer cases each year. I also found that roughly a half of people will get treated surgically, while slightly under 1/3 will get chemotherapy. So I've done some math and found out that roughly 5 million people each year will need to get some kind of chemotherapy. And if you take into account that this is not only a one-cycle treatment and there are many more people from previous years that are diagnosed with cancer, we can all agree that consumption of anti-cancer drugs is quite high currently in the world. But how do these anti-cancer drugs reach the environment? So first of all, they are produced in the factories, and shortly they are transferred to hospitals. There they are given to the patients, and we can say that modern oncological wards do have some kind of special waste and wastewater treatment plants or purification units, but it is questionable how many of hospitals that are not in high-income countries do have such special wards. So hospitals are one potential pollution spot. On the other hand, many of the patients are released after given the chemotherapy to their homes, and basically they are excreting parent compounds and transformation compounds, and all of these chemicals are then released into the sewage water, and apparently it is then transferred to wastewater treatment plants. According to the latest UN water report, as much as 80% of wastewater is not treated before it is released to the environment. So this is quite a big problem. The situation is somewhat better for Western countries, but still a lot of wastewater is released without previous treatment. So eventually, that is how we have cytostatic residues in our fresh water. At this point, I would like to say that such drugs are not persistent as pesticide DDT is, but those drugs are constantly released into the environment, and they can be classified as pseudo-persistent pollutants because they can be traced in the environment every day. And what is important for this part of the talk is that they usually occur in this nanogram per liter scale. So can these drugs that have license to kill have some impact on human health and the environment? That seems to be the question. To answer it and to tackle the problem, the Cytothreat project was initiated in 2011. The countries highlighted in yellow participated in this project, and the main aims were to develop analytical methods for better and more sophisticated detection of residues in the environment, then to assess the effects of what these drugs can do to organisms and humans. Back then it was quite novel to explore the effects of mixtures, and basically at the end of the project it was aimed to give some guidelines and risk assessment about cytostatic drugs. So let me jump straight into the middle of my talk. The first thing is how did we select which cytostatics will we assess? And I will limit my story to the case of 5-fluorouracil, or I will call it 5-FU. Here is the picture where we can see the top 6 cancer sites in the world. And here on the right side, given in yellow, we can see cancer sites that are prescribed with 5-FU as a therapy. So it is breast cancer, stomach cancer, colon cancer, and ovaries. We can say that it is a highly consumed drug. And here is its chemical background. And what I would like to emphasize is its modes of action. So basically, the molecule has ability to mimic uracil and thymidine. So basically, it incorporates on one way into the RNA molecule. It also incorporates into the DNA molecule, but it also has the effects on the inhibition of thymidylate synthase, so generating even more DNA damage. What was also important for us is that it is quite stable. We agreed that 80% of stability at given exposure temperatures for enough time is satisfactory. So at this point, we agreed that this compound is quite stable, it is highly consumed, and its price was acceptable to us, so we could proceed with further experiments. And the first experiments in line were those done in vitro. So we have performed analysis on target human cells, so HepG2 cells, cancer cell lines, then on the non-target cells, human peripheral blood lymphocytes, or more precisely blood cells, and finally the zebrafish cell lines as a representative of aquatic organisms. So first what we've done is we had to set up the concentration range to perform testing, and we have done that using MTT assay and acridine orange and ethidium bromide vital staining. And we got quite good results. The compound was not so cytotoxic at higher concentrations, but we wanted to limit the range more appropriately to environmental ones. So we have decided to use the range from 0 to 10 micrograms per milliliter. And the first genotoxicity assay we have done was a micronucleus test. And here we got only occasional positive results on target cells on higher concentrations. We had no results using zebrafish cell line or human peripheral blood cells. Moreover, we have done a comet assay, and similar no effect was observed for human cells and HepG2 cells, while there was some kind of dose-responsive increase in and significant increase in DNA damage, but again on higher concentrations. When we first got these results, we were both sad and happy. Sad on one side that we didn't get strong positive results that we expected, and on the other hand, we were quite happy because at this point we could say that even lower concentrations that are relevant for the environment could pose really small risks. And to get more information about the impacts of long-term exposure in aquatic organisms, we have done some in vivo tests using zebrafish model as a model and using these two-generation tests. So to briefly explain what does this two-generation means, it means that the zebrafish are actually under exposure since they are conceived. So the parents before spawning are exposed to the R5FU in this case, and then the fish hatches, and for the entire life they are exposed to the R5FU, eventually again spawning here for 4 times, and then generating this second generation. You can see here also that we have done some tests for fish old 4 months old and 7 months old, as well as for new generation of fish. I can now say that in the second generation we did not observe any changes in survival and body parameters. But let me now just talk in more detail about this first generation. So about the size of the fish, they were the same length in the control group as they were in the exposed groups, but we have observed some changes in their weight, and we believe that these changes were reflected by pathology and histology what we observed. So first I'm going to talk about the reproduction. Here we can see the results from the first spawning, where the exposure to this drug didn't have any effects. But when we saw the fifth spawning, there were also no changes. Here we can see only at one point a larger proportion of infertile eggs, but experts in breeding of fish told us that this is due to constant change of tanks and exposure solution in this quite sensitive period. And we can see that at the highest concentration, the numbers were quite similar. So we concluded that there was no reproductive effects. On the other hand, when we observed the histological samples from kidneys, we saw quite a large number of hyperplasia of hematopoietic tissue that is found in fish at the lowest concentrations, while at the highest concentration we also see severe damage to kidney structure. Presented here is one example of tubulonephrosis. Liver was also one target organ we explored, even at the lowest concentration, but more prominent here at the middle concentration. We saw adipose infiltration to hepatocytes. Those are these little white spots in the hepatocytes. And at the highest concentration, there were even signs of liver atrophy. When we looked more on the molecular level, we have performed some gene expression analysis and found that more than 1,000 genes were differently expressed at these exposure conditions. Most significant results were gained for the metabolic genes, mostly for the lipid metabolism, and overexpression was observed for oncogenes and for DNA damage repair genes. So this was quite interesting because using the COMET assay, we were able to see dose-dependent increase, and that was significant for the highest two concentrations for the liver cells. And speaking of genotoxicity, we also done some tests on the blood cells. And here again, we have done COMET assay that showed a significant result here for the middle concentration, but more straightforward results were gained for micronucleus test. Here we have observed dose-dependent and significant increase for all concentrations, even after 4 months of exposure, and then repeated analysis after 7 months. And taken together, we have concluded that this long time exposure to relatively low concentrations that could be found in the environment could have impact on aquatic organisms. At this point, I also want to go back to Rachel Carson and her quote that in nature, nothing exists alone. And this is unfortunately true for the cytostatics as well. So in the next step, we have done some mixture testing, and we have part of using 5FU, we have used also the etoposide, that is topoisomerase inhibitor, and also imatinib mesylate, that is firstly approved tyrosine kinase inhibitor. We have used again concentrations that are relevant for the environment, and we have exactly the same concentrations of each compound. When we run the genotoxicity tests, first one was the COMET assay, and we have observed a higher number of highly damaged cells, even at the lowest concentrations when these three compounds were in combination. These results were supported with the increased frequency of micronuclei, and we have also observed the higher frequency of sister chromatid exchange. What we also wanted to see here is whether some oxidative changes appeared during this exposure, and we have found that some kind of oxidative DNA damage was observed for the highest concentrations, and yet again On the level of lipids, we have observed the increase in concentration of malondialdehyde. So this product is also the sign of lipid peroxidation, but also it is one potent molecule that can be transferred to nucleus, and there it can form adducts with DNA, creating more DNA damage. So this is quite one interesting biomarker as well. Finally, we have observed more changes to proteins. So these are protein carbonyls, again, not from the lowest concentration, but the first next one, and we observed some kind of dose response. As a small conclusion of this part, I wanted to show you this little picture where it says that when we have performed tests on human blood cells using single compounds, we were actually not able to detect any DNA damage, but using only 3 of those compounds, we have managed to broken those thresholds and found positive results. As a reminder, as I said, these compounds are found in the environment in this nanogram per liter scale, and this is not entirely true actually. Our colleagues extended this work to real life scenario and found that these concentrations can be several thousand-fold higher. Here we can see results from, this is one town and this is another one, and these signs represent this is hospital effluent waters, this is wastewater influent, another one, and the last column represents wastewater effluent water. So basically, after the water purification, these concentrations are still, let's say, more than 10 times higher than as it is thought they are represented in the environment. And these wastewater were, of course, proved to be toxic for zebrafish cell lines, for aquatic organisms, and lastly, for plants as well. And I would like to stop here. If you found this talk and the topic interesting, I will recommend this link to follow. It is full report of the project, and all the results are here. You can also scan it with a QR code. It's very extensive, and it can be used both for research and perhaps teaching. And at last, I would like to emphasize one take-home message, and that is that cytostatic drugs are present in the environment and they have toxic potential. So this leads us to at least 2 future perspectives. One is that we really need to reconsider the use of non-targeted therapy, and that leads to development of novel chemotherapeutics. And on the other hand, what will actually be perhaps more easily to do is to develop more efficient wastewater treatment programs that will even more minimize the impact of these highly reactive drugs. At the end, okay, this is the hardest part. So as I promised at the beginning, I have pretty much thanking to do. So I would like to thank all the founders that supported our work during these years that can be seen on the right. I would like to thank Croatian Genetics Society, and of course Ana Maria and Mirta for writing this nomination letter. I would like to thank the EEMGS Society for presenting me with this award. I must say it again, I'm really happy and really honored to be in this selected part of awardees. And of course, I would like to thank these 3 people the most. So Vera Garaj Vrhovac, my mentor, who involved me with all her projects, and she was there at the early beginnings of my career. Then I would like to thank Goran, the best colleague there is, my postdoc mentor, and I believe the future Fritz Obels Award candidate. Finally, I would like to thank Anna Maria, who introduced me with all this oxidative stress story. And I must say that I didn't have to work for one day in my life, because this is like another scientific family that I switch between my home and work. And it was really a joy to work with you, and hopefully it will extend for many more years to come. There are also a list of people I have to thank, most of all Metka, Bojana, Matjaž, Jana, Robert, Akos, and the rest of the Cytotrack team. Then Mirta, Maya, our technician, who is very helpful, Nevenka, Višnja, Davorka, Božena, and the rest of the people in Croatia. And of course, I have to thank Danny, Isabel, Pep, Manuela, Karina, and the rest of the people from some coast actions that I was having ability to hang out and to collaborate. If I accidentally omitted somebody from the list, this is really not intentional. I really thank everybody who written one letter in all of my entire scientific career. I'm really grateful, and none of this would be complete without family and friends. Some of them you have already seen, so hi to them. They are watching at home. And at last, I would like to dedicate this award to my mother-in-law. She was very enthusiastic for my scientific research. She was also involved in some volunteer recruitment when we had some troubles, and unfortunately she passed away too young. And this one is for you, Marina. Thank you, and thanks everybody for watching. If there are some questions, please type them into the chat and I will gladly answer them later. Thank you.